So if you don't know me, I'm Greg Conradi Smith. I'm in applied science and biomath and neuroscience. And this is going to be a discussion about something that's an emerging research interest of mine, but it's also fairly pedagogical, which is, I tend to do that in these talks. So let's get started. So it's membraneless organelles, the Conradi equation for liquid-liquid phase separation, and my love-hate relationship with generative AI. Okay, so we all know what organelles are, right? So let's test ourselves. So which one of these does, I'll do an easy one, protein synthesis? Let's do an even easier one. Which one does cellular respiration and aerobic contains the DNA and direct synthesis of proteins and ribosomes?
Yeah, all right.
Yeah, and I think lipid synthesis is the smooth endoplasmic reticulum. Anyway, so you can, we all know what we're talking about with membraneless organelles, or membrane-bound organelles, or just organelles in general in eukaryotic cells. Now, as a person who models cell physiology kind of for a living, it's kind of amazing the extent to which people have focused on ordinary differential equation models, and they've worked extremely well. So like our course cellular biophysics and modeling is all about point neuron models, right? There's one membrane potential, and you have to take the sequel course to learn about dendrites, right? So the same's true of calcium signaling. There's a cottage industry of modeling cell signaling mediated by calcium, and so much of it is just like a variable for the cytoplasm and a variable for the ER concentration of calcium, and they work really well. But this is a little bit confusing and troubling when you can see like the complexity of the intercellular milieu, right, compared to the idea of a continuously stirred tank reactor, which is essentially what the ordinary differential equation models are assuming. So the real cell is spatially heterogeneous, compartmentalized. There's all these membrane systems. Different compartments are at different pH, right? There's different ionic concentrations, as we know from just the basic electrophysiology of excitable membranes, and from calcium signaling. Electrical potentials are different depending on what compartment you're in. And then what we probably don't think about very much, what we should think about more, is that things are extremely crowded in the cytoplasm and other compartments in the cell, so there's what's called molecular crowding. That's sort of the idea in this picture that so many of these macromolecules are packed in, right, together, that if you are a relatively large macromolecule, you're not going to be able to diffuse the way you would if you were a smaller thing. So there's just not enough nooks and crannies to get around if you're of any reasonable size. It's sort of thermal agitation is like a dominant kind of phenomenon. So like, imagine living your life in the midst of the biggest storm you could ever have be in. That's what's happening, right? All of the reactions are happening in the context of this incredible hurricane of activity. And of course, the cell's alive, right? So many of the processes are highly regulated and they consume ATP. Things are not at equilibrium. Equilibrium means dead cell, right? And the other thing is that if you take out a piece of, just say, the cytosol, so we really simplified it, right? We picked one compartment and kind of pulled it out. If you ask, like, what is that stuff? Well, it's more like in an egg, there's the white part and the yellow part. Which one's the yolk?
The yellow part.
The yellow part. So it's more like the yolk. That's the oocyte, right? So the stuff that's in the yolk is the best example of what cytoplasm is. It's gunk, right? So it's colloidal. It has gel or sol properties. What is a colloid? Well, a colloid is There's a lot of different kinds, but essentially it's when you have droplets of one phase of matter surrounded by kind of a ubiquitous state of another phase of matter. So we're familiar with like a gel is droplets of liquid trapped in a solid substrate. Foam is gas in a solid milieu, right? Emulsions are two different liquids, right, together, like oil and vinegar. When you shake it up, and it's an emulsion for a little while, and then it separates. So sols and gels are probably the best way to be, there's sort of a liquid aspect and a solid aspect. And, it's well known that things like the concentration of divalent cations influence very much the properties of gels, right? So when the calcium concentration goes up and then comes down, it's probably changing the physical state of the cytosol, right? It's not just a calcium concentration change in a solution, it's actually changing the properties of the gel, right, that is the cytosol. I think that that's gotta be true. I know it's true in the context of people doing physiological gel modeling, and there's swelling that can occur when you change the concentration of divalent cations. So what's a membraneless organelle? Well, it's the idea that this compartmentalization and heterogeneity, sort of functional specialization of different intercellular regions is not only membrane-delimited compartments, right? So let's take membrane systems and just say that's way too complicated, like the Golgi, right? I have no idea how you would ever model that, right? A membrane-delimited compartment, well, I've seen models of cells where there's the sort of ions crossing the various membranes, right, are involved. You might have cytosol, ER, maybe even mitochondrial compartments, right? Okay, so we can kind of handle the way that we would think about modeling membrane-delimited compartments. But a lot of the compartmentalization in cells does not have membranes around it, right? So here's a current review of 20 or so different membraneless organelles. So that's sort of the catchy term that people have started to use for all these specialized regions, a lot of them in the nucleus involved in gene regulation and expression, and also a lot like stress granules that are just in the cytosol. So I'm at the beginning of getting really interested in this, and so I am not ready to review all the different membraneless organelles and their distinctions, right? But I do have a pretty strong background in physical chemistry, and so the question of how do these membraneless structures sort of appear sort of as units that are separate from the overall gel-sol kind of milieu, right, is something that I thought I could pick up pretty quickly. And so that's what this talk is about. So the kind of generic picture here is that in, for example, the cytosol, you have these membraneless organelles, which are essentially regions where the concentration of something that's dissolved in the water, right, is going to be very elevated. It's the basic idea. So the border is sort of not really super well defined, but it's defined to the extent that the concentrations of various elements change dramatically at that one region. The simplest picture is where you've got the solvent and then sort of one polymer, right, macromolecule, and if these molecules have some sort of the right concentration and the right affinity for one another, then they'll segregate out of solution. They'll make little droplets of the polymer that are separate from what's going on in the external region. And in fact, if this happens, they might actually have client molecules that get attracted into that space, right? So you could, imagine that there's like a couple different types of those purple molecules, right? And the red ones are coming together because of some calcium signal or some other regulated process, like there's a phosphorylation event. Some post-translational modification of the polymer itself leads to going into this state, right? And now what's happened is that these client molecules are now concentrated, and perhaps at the higher concentration in the droplet, different biochemical reactions take place, or the same reactions at different rates, right? That's the sort of general picture that people think may be happening.
Is separation of oil and water an analogy?
Yes. It's I think I'm entirely focusing on the idea of liquid-liquid phase separation. So we're thinking of both regimes as being liquids, right? With different concentrations of macromolecules. All right, so that principle of liquid-liquid phase separation, so, phase is this one of these words that's wonderful because it has like 100 different meanings, right? We're not talking about phases of a cycle or an oscillation, or the phases of the moon. We're talking about phases of matter, kind of, except we know the phases of matter, gas, liquid, solid, plasma, right? This is not changing, this is not freezing or melting, so why do they call it phase, right? What's the phase? Well, there's first order and second order phase transitions, and this is an example of a second order phase transition. It doesn't take heat, right? There's no change in the latent heat required for the phase transition. So when you heat up ice, as soon as it starts to melt, you put your thermometer in and you know it's 0 degrees Celsius if there's a slurry, right? And it will stay 0 degrees Celsius in a continuously stirred tank reactor as you heat it up until all of the ice is water, and then it starts to get hotter again, right? So that's a great illustration of the difference between temperature and heat, right? But that's also a first order phase transition, whereas liquid-liquid phase separation is the second order phase transition, no heat consumed, or right, when it happens or required. So the way people think about it is that you just have two different states that have different sort of minima in the free energy. So that's a chemical potential, and this axis, if you think of it as how much of substance A versus how much of substance B is in the solution. Those could be two different proteins, for example. Way down on this end, so I guess this graph is, there's more A on the right side because it says Ca dense over here, and over here it says Cb dense, right? And as you move from left to right, you go into a situation where Cb is dense, so you basically just have a bunch of Cb in the solution, right? And then there's some sort of transition where Ca ends up, Ca sats for saturation. And so there's a point at which you will actually start, am I getting this right? You start to see these little droplets, right, of A in B. So there's less A and there's more B. And then you move this way and it's like 50-50, and you move this way and it's mostly B, right, in the background of A. So there's a concentration dependence of how the droplets in the surrounding media will be interacting. And then there's also this region that they're labeling as metastable, which is basically, how do we say this? This middle region is the region that we're talking about, where you will spontaneously separate the two fluids, right? And here, outside this curve, is the region in the phase diagram where you'll have the one fluid will be dissolved in the other, but they won't be separated. And you'll be mostly one or mostly the other. Okay? This metastable state is basically when you can have separation if there's nucleation events, right? But if there aren't nucleation events, it won't happen spontaneously. So the fancy physics word is that if you don't require nucleation, it's called spinodal decomposition, spinodal phase separation, and it's binodal if you actually need the nucleation. Yeah.
I'm sorry, just to clarify, when you're saying that the formation of the phase-separated organelle is independent of heat, is there not a entropic barrier that you have to cross to, form this densely packed thing in the?
So entropy is definitely a big part of what's going on, right? But there isn't enthalpy, right? There isn't.
Sure, but I meant, so in terms of you're organizing something that's, more ordered.
Right, and that's what you're sort of overcoming, exactly.
Okay, so how are you overcoming that?
The simplest picture is that you say substance A here has weak interactions between it, so that they, at some point at a certain concentration, will start sort of sticking together and be more likely to be close to one another. So that would be enough. So you would have to get to a certain concentration before that interaction would become apparent.
And the interactions are non-specific, so you're not necessarily forming a complex, it's just these proteins, the weak interactions just sort of happen.
They're weak interactions, yeah, and you're not forming a huge aggregate. Many of the systems are, we shouldn't think of it as globular proteins interacting. It's more intrinsically disordered proteins. So the sequences are very nonspecific, and there might be charged moieties that would be interacting electrostatically. There's the common sort of model that people are using is called ball and stickers, or cord and sticker model, or something like that. So you have aspects of the polymer where the only important thing is the entropy of it being able to move around, and then you have other pieces that can interact with the stickers on other like polymers. In that context, it's easy to get atomistic simulations or lattice-based Monte Carlo simulations to reproduce this phenomenon. Yeah.
Could I ask another clarifying question?
Of course.
So I did not read anything for this, so sorry.
You don't have to.
I do. So what is the difference, and I haven't dealt with this, but these membrane organelles are the biggest thing in development right now, or one of the big things. So what's the difference between those and just very large macromolecular complexes, which is what we used to call these things that were clearly identified in the cell for agents? And then, are both of these liquids then? That's what you're talking about here? So you're calling the membrane organelle a liquid?
A liquid-liquid phase
A
separation.
liquid. Not
That's one of the
everybody
dominant
does.
models.
Okay.
It might not be that in all cases.
Okay, because there is a lot of controversy over that too, whether that is defined as a solid, but
Yeah.
for the,
Like Sarah is interested in the postsynaptic density and the role of SYN gap and other scaffolding proteins. It seems that their function actually seems to be controlling the phase of the postsynaptic kind of the postsynaptic density. So that's there's recruitment of molecules into that postsynaptic density depending on how well various proteins are interacting with one another in the density. And that's thought to be an aspect of how you, of LTP and LTD right now.
But
So that would be a CAM kinase 2 mediated phosphorylation event, which changes something which changes probably the properties of SYNGAP, which then leads to a phase transition. Now, there's another model, right, would be this idea of a big macromolecular complex, a molecular machine, right, kind of thing, where you have all these interacting macromolecules, and they're crowded, they're close together, they could be passing substrates along from one enzyme to the next. That's another possibility. Is that what you're getting at?
well, yeah, some of the membraneless organelles are viewed as solid, so it's a liquid solid kind of not true, but phase separation. So when I'm seeing liquid phase here, that's like everything in the cell, that's continually going on. So it's not a membraneless organelle that is dictating a liquid phase transition only. This is much broader than what we call membrane.
Well, I would say it's probably the case that there's different aspects of phase separation that are occurring in different membraneless organelles. But what you said about this is the same as kind of everything else. The liquid phase separation, that's the particular phenomenon, because the concentration of the two molecules changes dramatically right at the interface. And that's not something that, that's something that is the rate of change of those concentrations, right? There needs to be a physical explanation for why that's actually happening. And it's the affinity, one possibility would be the affinity that the polymers have with one another.
Right. But, yeah, this is another word for cell biology to me. So, because that's happening continually a billion times. Sorry, go ahead.
So you're saying that there are no membraneless organelles, or you're saying there's not a discrete transition between what they call membraneless organelles and just the milieu?
Yeah, I guess for understanding what's being modeled and what people are going to investigate or look at as the mechanism behind it, because we know a lot about what dictates these coagulations or conformations of lots of macromolecules together. You know that. But I don't understand the difference between, okay, here's a membraneless organelle, but here are these liquid phase transitions and separations that are happening continually, thousands of places in time, places in that cell. So, but just in terms of a definition, what would we be looking at here? Is there a size? Is there a time element? That's what I'm not
Well,
understanding.
there's certainly, when you think about something, these two different phases, when it's a mixed phase, then the size of going from a molecule of type A to a molecule type B is extremely small, right? When something's in solution, right, the atoms are freely moving and they're very, very well mixed. And that's not the case when the liquid phase separation has happened. So the people use the word mesoscopic, right, which means the size of a mitochondria and maybe 10, 50 times smaller as the scale that we're talking about of these droplets.
Okay.
Are there concentration variations at smaller scale? Yeah, there's diffusion-driven concentration fluctuations. But anyway, can I go on?
Yeah.
Can I just really quickly ask you, what's the difference between these coming together in like a preon, for example?
So I don't know whether people are just saying this for get funding, but this process gone wrong is thought to be associated with these types of protein. It's thought to be part of the process of amyloid formation or prion. Yeah. So that would be the case where the interactions started to become more strong.
Yeah.
And so then you really have this thing that's more like solid that's come out of solution, and that's.
Like to break it up and use like detergent.
Yeah,
versus
yeah.
If this, you just change the concentration of the liquids, it wouldn't naturally change. Is that the idea with that?
Yeah,
Okay.
yeah. I think this is kind of related to what Margaret's saying too.
Yeah.
Like,
Yeah.
but
I don't wanna get off track, but to, but to say that postsynaptic density has this kind of, this temporally, it's so important for neurons to have a stable postsynaptic density and to regulate it in a way, and I never think of it as this sort of two things bump into one another, and if there's enough of them, some sort of change in the concentration or anything. I think it was a deliberate protein synthesis dependent with protein-protein interaction domains that makes a scaffold that's stable and strong.
yeah,
And so I don't.
I don't know yet.
Okay.
This is
Alright.
we're reading on it.
Okay,
Um,
I'm interested,
but-
like, I've never thought of it that way, and you're.
But people will mention that the presynaptic terminal also, that everything that we've been told about the fusion of secretory granules, right, also seems apparently to have. I mean, is it hype, and we're
Yeah.
just calling everything this now, or is it really the physical process that we're talking about going to be key to understanding the reaction rates? Or
Yeah.
the way that spatial things end up contributing, right? I don't know. I mean, like I said, I'm just at the beginning. Is it okay to?
You can keep going.
I'm not an advocate necessarily. I just think it's really interesting, right?
Yeah.
Okay. So now this is Margaret's gonna like at this point. It's so unrealistic, right? All right. So the way physicists deal with this kind of thing, this would be coming out of like polymer physics, for example, is they write down a partial differential equation that describes the demixing of the two fluids, right? And physicists tend to do this. This would be almost like a toy model, right? Where it's illustrating the principles, but there isn't going to be a representation of individual molecules here. It's a continuum model. And what the equation that people have been thinking about a lot since the 40s is this Kohn-Hilliard equation. It's not Kohn from Ratha-Kohn, different Kohn. I looked that up. Anyway, so this phi is a order parameter, and what that means in this context is just take a coarse grain take a picture at the mesoscopic scale of a little bit of this stuff, and is it mixed, or is it, like what percentage of type A versus type B is it, right? And if it's mostly type A, we'll call that 1, and if it's mostly type B, we'll call that -1, and 0 means perfectly mixed, right? So it's just a volume fraction, kind of. But it's an order parameter because the disorder state is at 0, and the -1 and +1, right, are the order states. M is a mobility that can depend on that parameter. Del of mu is the gradient of the chemical potential, and the phi and the chemical potential depend on space and time, right? And then this is like really important kind of continuum modeling stuff if people haven't run into it yet. But this equation is just called the continuity equation. It's telling you that there's conservation of mass, basically. So the rate of change of the quantity has to be dependent on the divergence of the fluxes in that quantity. And if there aren't reactions taking place, those add up to 0. So that's just conservation of mass. And then this is kind of like Fickian diffusion, saying the flux is proportional to the opposite of the gradient of the chemical potential. Okay? So
What about Fickian diffusion, is that what you said?
Fickian diffusion is when you say that the diffusion flux is the opposite of the diffusion constant times the partial of your substance times respect to x in 1D. And if you make the distinction between conservation conditions and constitutive relations, this is a conservation condition, this is a constitutive relation, that's a constitutive relation. It's something written down because empirically things appear to obey this when you're at the right concentration range and so on. So like Hooke's law for springs is a constitutive relation. But the fact that kinetic and potential energy, when you add them up, you get one number or whatever, right? That's the conservation. So the question then is, like, where is the free energy going to come from? And basically what's done, this is again the physicist's view of things, is the free energy. So maybe I should have used G here. This is G, Gibbs free energy. Basically, there's going to be a term which represents a potential, which is double well, so that the two states where you're demixed, right, the thing to some extent wants to be in those two demixed states, one or the other, and this term here is basically just penalizing gradients in the solution. So if you only had this term, then if you think of like something starting up here, half it's going to roll down there, half of it's going to roll down there, and you're going to just have A and B in different locations in your simulation, right? But now the gradient thing here, the square of the absolute value of that gradient of the order parameter, makes it so that there's smooth transitions, so that to go from A to B, there has to be a mixed region. Okay, and so this is the really cool part. You use calculus of variations to derive the free energy from the free energy functional. So derive the chemical potential from the free energy functional. So after you do that, you get this, which is different than that. This term is actually the derivative of that term. Relatively complicated thing that we're going to suppress for a minute, but I can go into it later, right? And then you end up saying, oh, well, that's the chemical potential, so I'm going to put that in, and if m's a constant, then the gradient of the divergence of the chemical potential is the Laplacian of the chemical potential. So this is the thing that people are calling the Cahn-Hilliard equation. It's a fourth order differential equation. There's fourth derivative in space. Now, if that notation with the del operators, right, doesn't mean anything to you, there is an entire semester of vector calculus, right, that you should take or watch YouTube videos on, right? It's really, really important. And the great thing is that I don't have to really be explicit about how many dimensions am I working in if I understand vector calculus, right? So it all starts with understanding how this del operator is a partial derivative of the function with respect to x and a partial derivative of the function with respect to y and any other coordinates times a little bitty unit vectors pointing in the different directions. And then if you know about vector calculus, you can derive everything that's on the board. And we're going to move on.
E is the unit vector in this case.
Sorry?
E is the unit
Yeah,
vector.
these are the unit vectors. Okay, so here is a simulation. So it's a 2D simulation, space in space, x and y. Time is the movie. And you're seeing the demixing, you're seeing the oil and the water, right? Occupying larger and larger chunks of space as the demixing occurs. Okay, here's another one. The difference here is that I'm starting out with only 30% of the yellow or something. And the amount of yellow and the amount of blue doesn't change. That's a conserved quantity. So you can be separated or mixed, but the total amount of phi in the simulation can't change because of that conservation equation. This process where the bubbles are getting bigger and bigger is called ripening. And you can even sort of see did you see how sometimes if two of them come together, then they end up wanting to become spherical, right? Well, that's happening because there's a penalty for any gradient in the system, right? And any place where there's an interface, there's a high gradient. And so the thing is going to try to make that surface area minimized, and a circle is minimizing the surface area. Now, if this there's aspects of this that aren't in here yet. It's important to understand that. Like there's right now no pressure, right? You can imagine a situation where there was an osmotic pressure, right? If these were sort of representing membrane delimited compartments, you would want there to be other physical forces in the free energy.
If you let that go for a much longer time, would it want to become one big one?
It'll ultimately separate completely. If you have the free energy the way that I wrote it,
Okay.
the standard model, ultimately it separates completely, but in time it gets slower and slower. So I think it's like cubic. You have to wait 3 times as long to get the same amount of change.
You mentioned this is modeling oil and water. Was that just an example?
Just an example.
Of what?
Yeah. It's just the first example of liquid phase
parameters
system.
that went into that. Okay.
You saw how many
I understand
parameters.
those.
There's no parameters in this
Yeah.
one. There's actually,
But this wouldn't
what are the
apply
parameters?
to a bunch of biological situations.
There's no, it applies to, it's very generically applicable for certain aspects, right? The only parameters in the system are this one, which I set to 1. It doesn't, that just scales how fast things happen. That's just time. And then there's this one, sigma, which is how penalizing are the gradients. And then you can have these wells. The wells can be at different depths, and I've done it so that they're at equal depths. So there are parameters here, and people have explored to death the differences.
So I think this equation is a mass conserving, so the total mass is also a parameter.
That's right. And the total mass, right, is basically the integral of phi over the whole system. And so that's why in this case, if I start with 30% yellow, it stays 30% yellow, right, throughout. It's a conservative system, yes. You can easily do 3D simulation. So these are isocontour lines. There's a little movie of the 3D stuff happening. All right, so I have, 10 minutes? 5 or 10 minutes. So, man, these questions, they really slow me down, don't they? So, how did I do this? You might ask, how did you get to be so fast? How did you do this? Why is this? Okay, I used ChatGPT, right? And I wanted to tell a little story about that. So basically, I started off with questions like, what is a function? Oh, never mind. Let's do, let's skip that one. Here we go. That was like 2 things that were explaining the modeling more and more, but I'm going to have to jump over that. So I started off with just a memory that if you're thinking about membranes or solutions separating, that there's something called a phase field model. And I'm like, oh, that would be interesting to know about. So I asked Chat, are phase field equations used to model membrane dynamics and or membranous organelles? And it says, yeah, I'm on the right track, okay? Then I said, please explain how, so Conn-Hilliard was on the last answer. Please explain how the Conn-Hilliard equations are used to model it. And it did a really good job of giving me the standard form of the Conn-Hilliard model.
You're being so polite, Chad.
Yeah, I always say please. I treat it well. And then it gave me applications, okay. And then I said, please provide MATLAB code that simulates the Conn-Hilliard equation in 2 spatial dimensions. And it said, sure. And it gave me the MATLAB code, and it, for whatever reason, decided to use a spectral method, so it's not finite difference. It's a spectral method. I was like, oh, that's kind of cool. Those, if you don't know what they are, you're sort of Fourier transforming everything, and then solving it, and then Fourier transforming back. And it worked perfectly. I was like, oh my God, I really need to start understanding how I could use this in my research, right? Okay. Then I started asking, well, I've heard that the Conn-Hilliard equations can be extended so that their solution represents the dynamics of a biological membrane with spontaneous curvature. Okay? So that's sort of the same type of equation can be used for membrane-bound organelles as well as like lipid vesicles and so on. So I'm asking it about that, and it says, oh yeah, that's right, you just put this term in here, which is kind of like Hooke's law, where the curvature, right, there's a spontaneous curvature, and that's the actual curvature. You square it, and that's the free energy functional. It's like, it's easy. And it said, here's your new MATLAB code. One of the few changes was this. It put in h minus h0 squared. And I went to run it, and it didn't work. Nothing happens here, lots of errors. So ChatGPT is starting to do worse because I pushed it a little bit, right? So I said, look, this doesn't work. More importantly, it seems to me that the quadratic spontaneous curvature term should be added to the free energy functional, not the chemical potential. The chemical potential would have a new term that was given by the functional derivative of this quadratic term. So I'm like, I know enough to say that is not right, okay? And then it said, you're exactly right. The quadratic blah, 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 and it gave me now this in the free energy, okay? And here's the new chemical potential, which is completely different than what it had said the first time, right? And then I went to run the code, and it didn't work. Okay, so at this point, I'm like, wow, how long did I, if the MATLAB code had worked, how long would I have been simulating right, this equation instead of this equation, right? Just because I believed ChatGPT, it was so confident in its answer. The way you get from, if you're interested in this, the way you get from here to here is a functional derivative. So you have to learn what functionals are and then variational calculus, right? So it's really cool stuff, but it's more than just taking the derivative. So now I've put this membrane, this bending energy in so that it's like there's a membrane between the two phases. And you can see what you're seeing here is that kappa parameter for this, how strong is the bending energy, how strong is the spring that keeps it at a certain curvature. That's one in all these simulations, and then what the spontaneous curvature is the gamma. So here in this simulation, it has some tendency to want to have no curvature. And so what you're seeing is, like, if you compare that to the other one, the regions are larger, right? And then I was trying to, just think through, well, how does this change when gamma becomes larger, when gamma becomes smaller? There's some effect, I don't understand it yet. Here's the case where it's in the bubble form, and you can see that what you'll end up seeing is that when gamma is negative, the bubbles want to be smaller. When gamma is positive, they'll be bigger.
Does the space that you're working in matter to how this runs? If it was a long skinny axon shape versus a square?
Right, the way the boundaries can definitely affect what happens, the boundary conditions. Everything that I'm doing is assuming periodic boundary conditions because that's the easiest thing to do to get something up and running, and because I had no scientific question involving boundary conditions. And the spectral methods also work really well when there's periodic solutions, so for all those reasons. So it's like simulating this on a donut, the surface of a donut, really.
So gamma equals 0, you said biases it towards straight line interfaces?
Yeah.
So then,
And then positive curvature versus negative curvature. Positive curvature is turning in towards the yellow, turning in towards where phi is positive.
okay. Oh, because you can see the little yellow in the top left, okay.
Yeah, I honestly, when it gets this, I sort of believe here, I wonder whether there's something going on with the mesh or something. I'm still, I'm not sure that everything's perfect here. This was just me trying to understand how adding this spontaneous curvature to a membrane at the interface would influence the solutions. Okay, so what do I want to? This is a good place to stop, I guess, since time is running out. I can do a couple different things. One I think I'm going to give a plug for Visual PDE, Um, So this is software that's running in the cloud, right? That will simulate two-dimensional and one-dimensional PDEs of all different types. And there's a manual available online, and it's really, really easy to use. Okay? So if you want to see the equations that are being solved, you just go up here. Those are the equations, right? And so I can't give you a complete tour, but just go to the site and play around, and you'd be like, if you want to have a reason to take Zhongping's partial differential equations course, you will, if you're not convinced, you will be after you do this.
It goes up to 4 variables, I believe, just in case.
Up to 4 dynamic variables,
Yeah.
and it doesn't go above 2 dimensions.
Right.
But like, what would be there? So they have the Cahn-Hilliard equation. This is they've added sort of fluid flow, I think, to it. So anyway, that's just think of that as another example, cool PDE. So I really think that this would be a lot of use to people. You know, you have, for example, Turing instabilities. You've got chemotaxis simulations. Something you should definitely use this in your class if you don't already, and so on. So
Ocean spills.
Ocean spills.
Shallow water equations.
I saw urban flooding.
From marine.
Urban flooding went by. Tracy
So basically, if you can you start with one of these that's close to what you want to do, and then you just start changing the equations and the parameters, and it's a really fast way to get up and running, at least in 2D.
And the publication related to this came out in 2023 in the Bulletin of Mathematical Biology.
So it's pretty stable too. I mean, it really doesn't fail too often. If you get a white screen, it's usually just your time step's too big. Right? So it's pretty good. Here's another thing. So in my love-hate relationship with generative AI, I wanted to share with you that Notebook LM, which is produced by Google, is a new app in the cloud. And what's different here is that you basically upload up to 50 publications or PDF files or links to the pages on the internet that you're interested in, right? Or links to Google Slides or Google Docs that you've written, right? And it will basically, the chatbot will interact with these publications, not with the whole internet, right? So when it answers a question, is the postsynaptic density a membraneless organelle? It's reading the 50 papers that I uploaded on membraneless organelles and coming up with an answer. So let's see what it says.
Let's see who said it.
What? What'd you say?
I said, and then let's see who said it.
Uh-huh. Oh, and so one of the cool things is that the postsynaptic density is a membraneless structure composed of protein molecules. Membraneless subcellular structures contribute to the. That's not very confident, right? But the great thing is it will actually tell me where it found that, right? So just give me one more question to show you how good this is.
How much water am I using to answer these questions? But that wouldn't be PDFs.
What are examples of membraneless organelles? So it's going to summarize what it gets from all of the papers here. So it'll probably come up with a list of 10 at least. What does this mean? What it means is I didn't have to read them all to look for this particular piece of information. It probably would have been better if I read them all, right?
Once you hit location, then you can go read
Yeah,
context.
yeah. So here's the overview. So here's the bunch of examples and the papers in which, they're found. So like, stress granules, reference 5, this is going to end up telling me where that actually came out of the abstract. So anyway, I found that this is actually a pretty cool way to work, and the other thing that it will do for you is it will make a fake podcast on whatever topic that you have. So this takes a minute to actually work, but let's see if I have membraneless organelles, notebook guide. So if you need to go, feel free. This is going to load the conversation. Here we go.
All right, so you sent us these papers on cell membranes and
So that's it.
phase transitions and then membraneless organelles. I mean, talk about a scientific trifecta. You're obviously somebody who likes to connect the dots between some pretty different sounding fields, which we love.
Yeah, it's that intersection of biology, physics, and even material science that we're going to try to attack.
Exactly, and we're on this mission to like pull out the most surprising insights from these honestly very dense papers.
Yeah.
So first up, allolioedel2024.pdf. And let me tell you, this is not your high school biology cell membrane anymore. This paper goes deep, deep into mesoscopic physics. You ever even heard of that?
It's all about kind of finding that sweet spot between like the micro chaos of all those atoms doing their thing and then the big picture, that macro world that we see, It's like, imagine trying to understand a cell membrane by keeping track of every single atom. It's like, trying to predict the weather by following every single air molecule.
Oh God, that's way too much. I can barely handle the weather app as it is.
Exactly. It's just not possible.
Okay, so this mesoscopic physics gives us a more manageable way to look at these crazy complex systems.
Precisely. And allioedel2024.pdf uses this approach to really model those cell membranes, but it gets cooler. They're bringing this thing called the Helfrich-Hamiltonian.
Okay, before you lose me completely, because I love a good intimidating scientific term,
Right.
Break it down. What is...
So, I'm showing the statistical physicist view when I put this equation.
I get that.
Okay, all right. So, and it's classical, right? It's from 1940s when people were studying polymers and magnets and things like that, right? That's where the equations first came out. And then people are saying, oh, this is also a model for liquid phase separation. There's good reasons from the theory of critical phenomenon why these equations should represent liquid phase. But it's not the only mathematical approach. And the approaches to get at the things you're talking about are much more complicated. There's something called lattice Boltzmann methods that would allow these more complicated interactions to be included. There's molecular dynamics calculations.
Oh, yeah, right.
There's sort of atomistic calculations where you have, this is the sticker region and this is the rest of the polymer and this is the sticker region. And so you can put in things that you think you know about the domains of the protein, right, and have them actually simulated. And so all these things are sort of out there, but that is like real research at that point, right? Because there's this whole emerging research area of like disordered proteins and
I wouldn't even say it's emerging. We know a
It's
lot
been out
about
there, right.
Yeah, I mean, and as people have studied those because those are the really
Yeah.
interesting proteins that regulate what's going on in
Yeah.
the cell
Yeah.
too. But
and so I think like a modeling the synaptic density, if that's something that truly seems like it's a reasonable thing to do, I think you'd want to do a calculation that
Okay.
allowed you to have the details of the protein structure in there. Not molecular dynamics level, but some level between molecular dynamics
exactly
and this.
using some of the
Yeah.
molecular dynamic because it's very informative to go to a more
Yeah.
macro level.
Yeah.
But even the physicists, when they were developing, there's a polymer and there's a polymer. They're not always going to be behaving the same.
Right.
based on the chemical interactions, you know, I
So
don't
the
care if it's
original
alive or not,
physicists'
you know.
work would basically, if you, there's a class of models called lattice models, where you explicitly have the polymer in 2 or 3 dimensional space, and the different monomers are occupying the various lattice points. And you could put, for example, a charge on one of them, or make certain moieties hydrophobic or
Yeah.
whatever. And then it's a Monte Carlo calculation, and they take much longer to run than this sort of thing. But you can then sort of study the thermodynamics of the aggregation of these different
Yeah,
polymers, right? And so that was like, you know, that's like, there's textbooks written about like how you specifically apply that approach to studying polymers. And so, yeah, there's a really, really interesting field, I think, that in terms of, for me anyway, doing the calculations in a way that will truly inform experiment and,
Right.
and not just be, this is illustrative, right? This is just
Right,
like
right.
illustrating the idea.
right. Anyway, yeah, I have a 1 o'clock, but that was really interesting. This is just such a big thing in development
Yeah.
now. And really trying to finesse, oh, there is a membraneless organelle, but it's really a continuum, and looking at how those are continually changing
Yeah.
in the cell
Yeah.
to make development work.
Yeah.
This was really interesting, Greg.
Right.
Can-
Well, let's. I
thank
mean, let's
you.
talk about it more.
Yeah,
I would love.
thank you. This
I mean,
was- this was
my
really cool.
like this is my 2 weeks of learning. Basically, is where I'm at, but I can already see that there's room for real research in terms of the computational side, because anytime you want to put realism into it, it becomes this big stochastic simulation that you have to do right.
So,
Um,
I was thinking about, the data that I have, which is mitochondria moving within this long radial process, and, I don't know if I'm gonna be able to say this, like, could you use your model to sort of describe what would happen if there was like random stochastic interaction versus the cell intentionally having fusion and fission? Would you be able to like discover what is intentionally happening versus what just happens because there's so many in the same compartment and they're bumping into one another randomly?
Yes.
Would you be able to do
Yeah.
that from this?
I would be able. I would be a great project actually. So, there are definitely models out there already of microtubule motor proteins moving things around,
Yeah,
right?
yeah.
The way that you handle the the diffusion random part is
Yeah.
clear, and then and then you just have to figure out how you represent fission and fusion, and like, does that end up? They grow, right? They
Yeah.
divide and then they grow.
Yeah.
So you're going to have to put something that's sort of like, they must have this in some of the chemotaxis models, where the bacteria actually divide,
Yeah.
right? So you have to like figure out like the way to do that. But I think something like that could be up and running pretty quickly. I mean,
It's
it would be a
really
good thing to do
like-
with a student, because that whole process of just figuring out the right equations is kind of the learning part.
I think that, like, I'm not really, like, a tool for a biologist to understand whether this is a cell's intention. You know, like, it's purposefully investing in protein moving in, like, the way that the organelle is moving with the microtubules, like, something like that.
Yeah, yeah. And so you like, yeah, so like what people will do, for example, if you take the diffusion equation and then you solve it, you can start with a Gaussian and then you plot its width changing as a function of time. But
Yeah.
that's something you can do experimentally, so you
Yeah.
can back calculate the diffusion coefficient,
Okay,
right?
yeah, yeah.
So you would be doing the same sort of thing, where you would say, okay, under the assumption that there's no directed movement here, what would I expect to be happening? You figure out some way to quantify that in the experiment, look at it in the model and see if it's the same. And I think that that's a really cool use of the modeling, because you're not just trying to reproduce a phenomenon, you're trying to back calculate, like you're looking at the experiment going, is this consistent with the following microscopic picture? I think that's a neat way to use this type of modeling.
So when it comes to synapses, if your student's going to keep going down this road, I wonder if it would be useful to look at the papers of Carl Svoboda's lab and I think Ryohi Yasuda,
I know.
right? Because they were modeling like what can get into a synaptic, a spine, and they had a lot of like the physical parameters fit and how much it can spread, and using what I think is like top of the line microscopy in order to model how things move in and out of spines.
That's a really good suggestion.
Um,
So I've heard him speak. I
yeah,
really like him.
I mean, he's a
actually.
rock star.
Um,
Yeah.
That's a great idea.
Yeah, because I mean, they were particularly interested because LTP needs to have spines need to be controlled independently, and you can't have just anything washing into them if you want to have real changes that are meaningful for neurons. And so they want to
How do you
is that
keep
the
things
case?
compartmentalized?
Can
Yeah.
kinase move in? What sort of that sort of stuff.
Yeah.
And
And there's
then
little bits of ER in there too. I've
yes,
heard.
and that can,
That's kind of cool.
I know, and possibly local protein synthesis. Another person that I really respect is Kristen Harris. She does EM work, and she will talk about like ribosomes being near synapses that have been potentiated, and what that might mean for local protein